Hey guys, I am Palec from STEM 1205. In this video, I will show you how to make my 3D bacterial cell. For the materials, you'll need carton, yarn, scissor, glue, construction paper, and foam tape. Bacteria are prokaryotic, generally unicellular organisms which exist as single cells or as cell clusters. They are among the organisms that are too small to be visible to the naked eye. Thus, they can only be seen with the aid of a microscope. An exception to this, Margarita and Epilopicium, which are invisible without a microscope. Presence of bacteria may be considered ubiquitous. They are easily the most abundant of microorganisms and are present virtually everywhere. Bacteria grow in a multiplicity of environments ranging from hot sulfur springs to deep freezers, from high to low acidity and high to low osmolarity. In addition, they can grow in both nutritionally rich and nutritionally poor situations. Hence, although each organism is uniquely suited to its own particular environment and rarely grows out of it. Now, I will show you the parts of bacterial cell and its function. Capsule. The bacterial capsule is a large structure common to many bacteria. It is a polysaccharide layer that lies outside the cell envelope, and it thus deemed part of the outer envelope of a bacterial cell. It is a well-organized layer, not easily washed off, and it can be the cause of various diseases. This is the cell wall. The cell wall has multiple functions during bacterial growth, including maintaining bacterial cell integrity and shape as well as resisting internal pressure. Furthermore, it must remain flexible to accommodate the remodeling that is required for cell division and the growth. Plasma membrane. This is the plasma membrane. The plasma membrane, also called the cytoplasmic membrane, is the most dynamic structure of a prokaryotic cell. Its main function is a selective permeability barrier that regulates the passage of substances into and out of the cell. Plasmid. This is the plasmid. A plasmid is a small circular double-stranded DNA molecule that is distinct from a cell's chromosomal DNA. Plasmids naturally exist in bacterial cells, and they also occur in some eukaryotes. Often, the genes carried in plasmids provide bacteria with genetic advantages such as an antibiotic resistance. Cytoplasm. This is the cytoplasm. The cytoplasm or protoplasm of bacterial cell is where the function for cell growth, metabolism, and replication are carried out. It is a gel-like matrix composed of water, enzymes, nutrients, waste, and gases, and contains cell structures such as ribosomes, chromosomes, and plasmid. Nucleoid, DNA. This is a nucleoid. The nucleoid region is a regularly shaped section of a prokaryotic cell where DNA is housed. It lacks the membrane that is found around the nucleus of eukaryotic cell. In addition to DNA, the nucleoid may also contain RNA, proteins, enzymes that can be used for cellular processes. Ribosomes. These are the ribosomes. Ribosomes are microscopic factories found in all cells, including bacteria. They translate the genetic code from the molecular language of nucleic acid to amino acids. Proteins are the molecules that perform all the functions of all cells and living organisms. Pilus. This is the pilus. A pilus is a thin, rigid fiber made of protein that protrudes from the cell surface. The primary function of pili are to attach a bacterial cell to specific surfaces or to another cells. Flagellum. This is the flagellum. Flagellum is primarily a motility organelle that enables movement and chemotaxis. Bacteria can have one flagellum or several. They can be either polar, one or several flagella at one spot, or peritrichous, several flagella all over the bacterium. That's all for this video, and I hope you've learned some lessons about bacterial cell and its function. Bye!